Hello.
Sir, good morning.
Hello, Nitish sir, good morning.
Sir, I am muted. I cannot.
Hello. Can you hear me?
Hello. Can you see me, sir?
No. Hello.
Dr. Damodar. Good morning, sir.
Sir, good morning, sir. You're audible. We can hear your voice.
Yes, sir. Yes, sir. Okay, thank you. Thank you. Good morning to everyone.
Yes, sir. Good morning.
Hello. Sir, Nitish sir.
Yes, sir.
Good morning, sir. This is Dr. Anbu from Rama University.
Morning, sir.
Yeah, I think this is the time to start.
Okay, so can you please guide me how to share the presentation?
1 second. You have your PPT, you know.
Sir, good morning. Nitish sir.
Good morning. Yes, sir.
Yes, sir. In the
At the
top side,
uh,
there is a name called as share. In the
Share. Yeah.
share
Yeah,
is
sir.
the right. Yeah, click on that share.
Okay.
Screen is the
So I
right.
can share. Okay. Okay. I can share. Okay.
Can you able to do it, sir?
Yes. I'll just do it. Sir.
Yeah.
Uh, I hope everyone can see the presentation right now.
Yeah,
Yes,
sir.
sir, yes, sir.
Can
Now
you
it's
make it as a
visible.
full screen?
I'll make it full screen.
Yeah.
Is it okay?
It's not showing as a full screen. It's
I'll
not showing
make
as
it
a
full screen.
no, sir. It's not showing as a full screen.
I have made it full screen.
Can you change the slides? Just move up and down, sir.
Okay. I'm moving it. I'm moving the slides up and down.
No, slides are not moving,
Just,
sir. I think hope you have not shared the complete screen. Go to share. There's a for screen is there, right, sir?
wait, sir. Just.
There is a term called a screen, no, sir. Two things you can see. One is screen, and another one is Windows.
Yeah. I should share the entire screen.
Yeah, exactly. So that whatever the things
Okay.
you are doing, no, it will get changed.
Okay. Now, I think.
Yeah.
Yes.
Yes.
You put it in
My
full
entire
screen
screen
mode.
is visible.
The screen is visible, but it is not in full screen.
Yeah.
Yeah,
Now
now
it's fine. Perfect.
Perfect now. Okay.
Oh, yes,
Okay.
yes.
So, should I start?
No, wait, sir. 2 minutes and a few more people to
Okay.
join.
Okay.
So just 2
Okay,
minutes.
sir.
I will inform you.
Okay, sir. No problem.
Hello, Nitish sir.
Yes, sir.
So I think it's time to start. People have joined.
Oh,
Let me start with some introductory note with our peoples. Okay.
okay, sir. Okay, sir.
Right.
Good morning, one and all. On behalf of Department of Pharmacology, Faculty of Pharmacy, Ramaiah University of Applied Sciences, I, Dr. Manisha Devi, extend a warm welcome to all of you for the webinar session Stress-induced Neurodegeneration. It's my privilege to introduce to you all our eminent speaker, Professor Nitish Bhatia. Professor Nitish Bhatia is an accomplished pharmaceutical science researcher and educator based in India. He earned his PhD in pharmaceutical sciences in 2015 and has over 15 years of professional experience. Dr. Bhatia's research focuses on neurodegenerative disorders, neuropharmacology, medicinal plants, and toxicology. He has published extensively with 25 research articles, 15 review articles, 5 books, and 5 patents, and over 50 publications overall. An award-winning researcher, Dr. Bhatia has won multiple Best Paper and Presentation awards at national and international conferences. He serves on the editorial boards of several scientific journals and has reviewed for several other publications. Currently a professor at Vishwakarma University, Pune, Dr. Bhatia has previously held academic and research positions at institutions including Punjab University, Jaipur National University, Sri Sai College of Pharmacy, and Khalsa College of Pharmacy, and GD Goenka University, with a strong track record in both research and teaching. Dr. Bhatia has established himself as a leader in the field of pharmaceutical sciences in India. His work has contributed significantly to the understanding of medicinal plants, neuropharmacology, and drug toxicology. I now welcome Dr. Bhatia for the session.
Thank you very much, ma'am. Thank you. Uh, First of all, I thank my colleague, Mr. Damodar Naik, for calling me for this webinar session. I also want to thank Dr. J. Andrew and other faculty members of Ramaiah University of Applied Sciences for giving me this opportunity to share my insights, my research insights with you. Talking about today's session on stress-induced neurodegeneration, which I'll be talking in details in a few minutes from now. I would first like to introduce this topic to you, the stress and neurodegeneration. We all have been considering for so many years that neurodegeneration is a disease, a condition associated with old age. What we forgot to consider is the pathophysiology of this condition. A condition which is associated with old age can also come up in young age if the pathophysiological mechanisms, they hasten up very quickly, and if the toxicological profile of the body is high. So stress does all these. It increases the toxicological profile of the body, it increases the free radical content, it increases our capacity to get affected by various environmental toxins or day-to-day toxins. So by these words, I'll just start with this topic of stress-induced neurodegeneration by the permission of all the dignitaries over here. So this presentation will cover the introduction to stress, and then we're going to understand the stress, just a few basics of neurodegeneration, just a few. And then keeping in mind that there might be students over here, I have used a layman's language, not tried to be too technical over here. And I'll tell you what stress does to the brain and what are the mechanisms actually involved in making the stress cause neurodegeneration. And I'll tell you something about with 2-3 main neurodegenerative diseases on which my lab has been working previously, and what are the clinical evidences all over the world that have been collected which show that stress can result in neurodegenerative disorders. I'll also tell you what impact does stress has on the brain structure and function. And most importantly, at the end, I'll tell you how can we prevent and manage the stress? What are the current research works which are going on and what are the future directions? And finally, for the purpose of management, also for the purpose of senior management sitting over here, and for the purpose of all of us, I'm going to tell you what implications the stress has on the healthcare and the society. So the purpose why we are sitting over here in this webinar, looking at how stress causes neurodegeneration, is to delve into the intricate relationship between stress and neurodegeneration. See, stress is a day-to-day phenomenon. Stress happens to everybody. We are all stressed. Stress is, I should say, it's a necessary evil. Stress is required because with the help of stress, we look out for work, we look out for food, we look out for everything. But when stress becomes chronic, unpredictable, and more than what we can handle, then it results in pathophysiological phenomena, which can result in irreversible damage to the neurons. So I'm going to tell you the relationship between the stress and neurodegeneration, shedding light on the mechanisms, consequences, and broader, in a broader sense, I'm going to tell you the implications of this connection between stress and neurodegeneration. I'm also going to tell you about the biological pathways through which stress influences neurodegenerative disease, hastens them, increases their rate. So we aim to enhance the awareness and understanding of this critical health issue. So the knowledge which I'm going to give you, it may equip you with tools to explore the prevention and management strategies against stress, so that none of you ever in the life, encounters any neurodegenerative disorders. So let's start. So can stress degenerate neurons? First question. Stress is something which we encounter every day, and neurodegeneration is something which we consider to be a disease of old age, maybe after 70 years, 75 years of age. So stress-induced neurodegeneration is a very captivating title which I have kept. It shows an intersection in the field of neurosciences. Stress, everyday phenomenon, body working against stress every day, whereas neurodegeneration is a chronic phenomenon. So the intricate relationship between the stress and deterioration of neuronal structures and deterioration of neuronal function. So it has always drawn the attention of researchers worldwide. They try to think that, okay, day-to-day stress, day-to-day breakdown of the neurons, day-to-day inflammatory pathway activation, can it result in long-term complication known as neurodegeneration? So this phenomenon, rooted in the complex interplay of biological, psychological, and environmental factors. There are biological factors which affect stress. There are biological factors which can initiate neurodegeneration. There are psychological factors which can, somebody may be psychologically more prone to stress. Somebody may be psychologically more prone to neurodegeneration. So there are both psychological, biological, and environmental factors playing their role in both stress and neurodegeneration. But is there any link between the two? So there are profound implications of chronic stress on the neurological health. So if there are profound implications of chronic stress on the neurological health, it may inadvertently result in neurodegeneration. So we are in this presentation, I'm going to take you on a journey in understanding the mechanisms, consequences, and potential preventive measures associated with stress-induced neurodegeneration. So let us understand stress. I'm going to keep things very simple in a layman's language, so that I'm not going to talk about any complicated terms over here, so that all of us can understand this phenomenon. So stress is a word that requires no introduction. So there should be no introduction to this presentation. A word that requires no introduction, for its presence is known to all. It weaves its way into our lives. From the very first cry of our life when we were born, to the last breath of our life will be under stress. So stress demands attention. It affects our health. It affects our well-being. In this modern world, stress is a constant companion. It is there with everybody. Stress urges us to explore. You know, the first human being which came to earth was stressed because of hunger. So due to this hunger stress, he went to explore something to eat. So stress also leads us to the phenomenon of homeostasis. So whenever there is stress, our body reacts in a way so as to again come back to the balanced situation of homeostasis. So understanding stress is the first step towards a healthier and more peaceful existence of mankind. We should know what stress is. It is going to stay with us for lifetime, but we should know how to cope with it. So biologically, if we see, because we are all biological scientists, so biologically stress is defined as a psychological, physiological response that occurs when an individual perceives a threat. Threat could be hunger, threat could be enemy, threat could be a predator, or threat could be an injury. Whether real or imagined, there could be a real threat in some people, people suffering from depression, people suffering from anxiety. It could be an imagined threat also. So this threat response, this stress response, or I should say a threat response, triggers a complex series of events in the body, primarily involving the release of stress hormones, the culprit hormone, cortisol. And this cortisol is a long-term stress response hormone. The short-term stress response hormone, which actually prepares us for fight or flight, is the adrenaline. So these hormones, cortisol and adrenaline, prepare the body to respond to the perceived threat, leading to various physiological changes, including increased heart rate, blood pressure increase, there is heightened alertness. This, which we call fight or flight response, actually is an adaptive mechanism. It is there in every animal of the world. Okay, it helps the animals or individual, we should say, to cope with the challenging situation. However, if the stress becomes excessive, chronic, everyday phenomenon, unpredictable phenomenon, we don't know. I'm going to office today and I don't know which type of stress I'll be dealing with. Then it has detrimental effects on the body, because then it is going to increase the cortisol level in blood for a very, very long period of time. And we all know, being pharmacy professionals, we know that increased amount of cortisol for a long period in the body is going to have detrimental effect, including its effect on neuronal structure, function, physiology, anatomy, neuronal cell size, and ultimately it is going to lead to neurodegeneration. So stress can broadly be categorized into two main types. Acute stress, you can see a small, a very frightened face over here, acute stress and chronic stress. In acute stress, it's more of an excitement, more of an anxiety, more of like, I want to fight or I want to flight. But in chronic stress, you can see a face over here. In chronic stress, we don't know what to do, how to cope with it. And when we don't know how to cope with it, it is when the problem starts. So acute stress is for short time. It has temporary value. It is very intense, but it is for a temporary system. So in acute stress, more and more adrenaline is involved. So adrenaline causes flight or flight response, in which our body either fights with the situation or runs away from it. So acute stress is an adaptive phenomenon. It is there in every individual. It is there in every animal of the world. It prepares the body to cope with specific challenge. And to a certain extent, some amount of acute stress can enhance the focus and performance. We are watching a cricket match, and there are 12 runs to be taken in last over. So this is going to produce a small amount of acute stress. In adrenaline rush in our body will be at the edge of our seats, but once the match is over, once there is a decision whether we win or lose, after a certain period of time, we come back to normal. So there is a recovery phase. After the stressor is removed or resolved, the body typically returns to the normal state. But in chronic stress, as you can see over here, there is a person with loads of files, ringing phone, buzzing computer. There is a coffee, but you can see the face of the person. So the person is suffering chronic stress. Chronic stress is a long-term, persistent phenomenon. It has less intensity, but it has long duration. Chronic stress can result from ongoing continuous stress, work-related pressure, family pressure, financial pressure, relationship problems, health problems. So there is a physiological response, a slow physiological response, but more detrimental, more jeopardizing physiological response. Our body tries to adapt. It is not that our body does not tries to adapt to stress, but adaptation to chronic stress is very difficult. Why? Because majority of the phenomena associated with the homeostasis, associated with adaptation, they are diminished by cortisol in a long run. In a long run, cortisol diminishes our body to adapt with the stress. Initially, it helps us to adapt against the stress, but on a long run, it diminishes our immune system, it diminishes our body response, it diminishes our cellular response. Ultimately, it can contribute to neuronal damage and neurodegeneration. Then last phenomenon is the recovery. So the recovery from chronic stress often requires lifestyle changes. We need to take out the chronic stress from the root. Then only we can understand how the chronic stress can be dealt with. It is not that our body is going to produce a response against the chronic stress. It is that we need to change our life and lifestyle. Then only we can work against the chronic stress. So stress, if we see physiologically, it triggers a range of physiological and psychological responses. Now, I just want to tell some people about this HPA axis, those who are not aware of it. See, stress activates the hypothalamus directly to release corticotropin releasing hormone. Hypothalamus releases corticotropin releasing hormone, which acts on the anterior pituitary to release a hormone ACTH, adrenocorticotropin hormone. ACTH is released from the pituitary gland, anterior pituitary. This ACTH comes into the bloodstream and acts on the adrenal medulla, adrenal medulla, part of the adrenal gland, to release cortisol, to enhance, to double or sometimes triple the production of cortisol, and this cortisol is released into the blood. Now this cortisol is the ultimate stress hormone. So stress triggers a range of physiological and psychological responses in the body and mind. These responses are known as stress response. Our body under stress does not remain the same as it was normal. So what changes that are produced in the body, they are called stress response. So physiological responses to stress. You can see over here, there is a term stress response. There is, under the influence of cortisol, there is activation of autonomic nervous system. There is a release of stress hormone. Stress hormones is release is triggered by activation of HPA axis, hypothalamo-pituitary adrenal axis, resulting in release of stress hormones, cortisol and adrenaline. Under the influence of adrenaline, there is increased heart rate. If there is increased heart rate, there is constriction of blood vessels, and ultimately there is elevated blood pressure. Then the pupils get dilated, allowing for better vision. So I've told you stress might be due to a predator. So in order to work against a predator, animal requires better vision. So there is dilated pupil. There is increased respiratory rate to provide better supply of oxygen so that an animal can fight against the predator. There is profuse sweating to keep the body temperature low. And there is release of glucose. Now, this is one of the main factors associated with stress-induced diabetes. Cortisol decreases the production of glycogen. It increases the blood glucose levels in the body. So the body releases glucose into the bloodstream, provides immediate energy for the muscles, But this immediate energy is required only in acute stress, when we need to fight against a predator, when we need to fight, run away from a situation. What about chronic stress? In chronic stress, there is increase in glucose level, but that glucose is not being utilized. So it is causing diabetes. It is causing insulin resistance. There are digestive changes, stomach discomfort. I should tell you that cortisol also affects the release of leptin hormone. Leptin is one of the hormones of our enteric nervous system, which produces satiety. So if there is any student over here or even the faculty members, they must remember. So when we were students, when we had exam, just prior to the exam, nobody had any feeling of hunger. Our mothers used to say, have something before going to exam. We used to say, no, I'm not hungry. You were requiring glucose, but actually the stress level increased the glucose level. So just after completion of the exam, just when the stressor is over, everybody was, they rushed to canteen. Why? Because now after the stressor is over, after the effect of stress response is over, there is a fall in the glucose level. So we used to rush to canteen. So this is how digestive changes take place. Now I'm coming to this important part, this neurodegeneration. I'm not going to delve into the details of neurodegeneration, the pathways. I'm just going to talk about neurodegeneration. Neurodegeneration, as the name suggests, before I move to next slide, as the name suggests, degeneration of neurons. Degeneration means whatsoever the structure right now is, it diminishes, it goes into disarray, it shrinks, and it becomes non-functional. So this, when happens with neurons, it is called a neurodegeneration. So neurodegeneration is a process characterized by progressive. When it starts, it goes on. And irreversible degeneration, dysfunction, loss of function, loss of neurons, loss of nerve cells in the nervous system, particularly in the brain and the spinal cord. So this process is often associated with the development and progression of various neurological disorders, which I'm going to talk about. For example, Alzheimer's, Parkinson's, Huntington's, ALS, amyotrophic lateral sclerosis, and there are other diseases also. I'm going to talk about these three in much detail: Alzheimer, Parkinson's, and Huntington's disease. So coming back, stress and the brain. So how stress affects the neurons? Let us see in a bit more detail. So when we are under stress, cortisol is released. So stress release triggers the release of stress hormones such as cortisol and adrenaline. Under the influence of chronic stress, elevated cortisol levels can be damaging to the neurons over a period of time. So cortisol can disrupt the balance of neurotransmitter. We all should know that neurons, they do not just function as generators and propagators of action potential. Neurons also, they are involved in maintaining the redox potential of the brain. They are involved in maintaining the redox of the brain, reduction oxidation potential of the brain. So under the influence of cortisol, glial cells, a typical type of cells involved in the housekeeping functions of the neurons, they lose their function. They reduce in their function. So under the influence of cortisols, neurons, they go subtle death, a small amount of death every day. How? Neurons, actually they are involved in current production. Current production, that means the ion exchange is their major function. This ion exchange produces small amount of voltage discharges, voltage changes every day, every second of our life. This can result in activation of various proteins, various genes, which are silenced by glial cells. But if this silencing phenomenon of various pro-inflammatory genes is not there, it can result in inflammation over a period of time. And this we term as neuroinflammation. So balance of neurotransmitter is lost, balance of neurotransmitter is disrupted, which can result in interfere in the functioning of neuronal receptors. Then chronic stress can lead to a low level of inflammation in the body. I have told you, chronic stress affects the function of glial cells. Glial cells do the housekeeping function. Glial cells prevent the pro-apoptotic cells, pro-apoptotic genes to function. So if there is less amount of glial cell protection under the influence of cortisol, so then inflammation is bound to happen. So inflammatory molecules which are produced, I'm going to talk about one of them. One of them is nuclear factor kappa B, NF kappa B, which you can see over here in this slide. So these inflammatory molecules, my students are working on NF kappa B. These inflammatory molecules can harm neurons and can interfere with their normal function. One other phenomenon which many researchers have focused on is the oxidative stress. Neurons, they require high amount of energy. High amount of energy means that high amount of ATP. High amount of ATP means mitochondria would be continually functioning. When the mitochondria are continually functioning for the production of ATP, there is high production of free radical species or species with a lone pair of electrons. Under the influence of stress or stressors, or under the influence of cortisol, this protection of the mitochondrial membrane is lost. Mitochondria becomes more leaky, and under the influence of leaky mitochondria, these free radicals are released into the cellular matrix. This results in oxidative changes, peroxidative changes in the neurons, and can result in neurodegeneration. So there is neurotransmitter imbalance. There are changes in the brain structure. This is very important. Prolonged or severe stress can lead to changes in the structure of the brain. There is a particular area in the brain which is responsible for memory formation and memory processing, hippocampus, a region critical for memory and learning. So chronic stress can result in loss of dendrites in that particular region and can even result in death of the hippocampal neurons. There is particularly an area known as CA1 region, CA1, which is responsible for memory, recent memory, and this recent memory is lost. Before I move further, I would like to take an example. If you are under stress, usually what happens, you are having a key, you are having the key of the house in your pocket, and you forget that that is in your pocket, and you keep out looking for it. This happens. The thing might be there in your pocket. You might have seen that thing recently, but you forget about it. This is how stress affects the memory. This is how stress degenerates the hippocampal CA1 region neurons. The neurons involved in the recent memory, three-dimensional memory. Okay, so it also affects, stress and cortisol also affects the neuronal communication. So you might say, I might have seen this thing, I might have seen this person, but I'm not remembering what is his name or where have I seen this. Why? Because neuronal communication, a new neuronal communication process was not being done under the influence of stress or under the influence of cortisol. Ultimately, there is neuroinflammation, which can result in neurodegeneration. And I might tell you that this cortisol, being a steroid, has the property even to alter the genes responsible for protection against various types of pro-inflammatory markers, against various types of cancers, against various types of pro-neoplastic genes. And that is why people under stress, people who are constantly under stress, they are more prone to all these diseases which I have talked about, they are more prone to memory dysfunction, they are more prone to diabetes, they are more prone to even certain types of neoplasms. So let us see things in a bit more detail, just a bit more detail. So the process of hypothalamic activation, you can see over here, this is normal stress response. There is stress, hippocampus, the hypothalamus releases the ACTH, CRH, and then it acts on the pituitary, it releases ACTH, it acts on the adrenal gland, releases cortisol, and this blocks the inflammation. But under chronic stress, huge amount of cortisol is released. There is no negative feedback inhibition. So continually high level of cortisol, even the immune cells, they become resistant to the effect of cortisol. So there is persistent inflammation. Although cortisol is considered to be anti-inflammatory in nature, but when this cortisol remains in our blood for a longer period of time, just as in the case of there could be insulin resistance, there could be a cortisol resistance also. And due to this cortisol resistance, there is a feedback increased inflammation. Although it should have been low inflammation under the influence of cortisol, but in cortisol resistance, there is increased inflammation. So much so that it affects the hippocampal neurons. The hippocampal neurons, they die. They undergo apoptosis in the influence of cortisol resistance. So what happens when there is stress? There is a perception for a stressor. We know that, okay, time is sweating over here because the time is running out to complete the test. Then our
Hello. Hello, sir. You are not audible, sir.
Sir, actually, sir is not in the meeting. I think he left the meeting.
Oh,
By
sir.
mistake.
Am I audible?
Yes, sir. Yes, sir. You are audible.
Okay, I'm sorry that my call dropped. I'll just try to share again.
Yes, yes.
Okay. Uh, can you tell me on which slide I was?
When the cortisol, that mitochondrial mechanism he was explaining, sir, previously.
Okay, sir. Okay, sir. On this slide,
Yes, next, the three pictures with mechanism.
this one, sir.
Yes, and you can continue from here only, sir.
I can continue from here. Okay, no problem. So, biological mechanism
Can you
of
please
stress.
put it in full screen mode, sir?
It is in the full screen mode, sir. Is it not being shown in the full screen
Yeah,
mode?
yeah, it's fine, sir. Fine,
So, yeah,
sir.
yeah.
Okay. So biological mechanism of stress mediated neurodegeneration. So I'll just going to talk a bit about the biological mechanisms. So see, because time has also elapsed, so I'll be bit quick. So cortisol mediated reactive oxygen species production. So stress causes release of cortisol. Cortisol causes deterioration of the neuronal function in 2-3 mechanisms, with the help of 2-3 mechanisms. I'll talk about first mechanism. First thing, cortisol increases the reactive oxygen species production in the neuron. So cortisol, which is the primary stress hormone, can indeed contribute to the production of reactive oxygen species. You know that cortisol activates an enzyme, I'll highlight over here, nicotinamide adenine dinucleotide phosphate oxidase, NADPH oxidase. So cortisol activates NADPH oxidase, and NADPH oxidase is responsible for producing a free radical, superoxide free radical, which is the primary reactive oxygen species. We can call it the mother reactive oxygen species also, because this superoxide free radical can serve as a precursor for the generation of other reactive oxygen species such as peroxide, such as nitrous also. So when cortisol binds to its receptor in the cell membrane, it activates NADPH oxidase. NADPH oxidase results in an increase in the superoxide production. So next is cortisol mediated antioxidant depletion. We all know that our neurons, just like any other cells, have got antioxidant mechanisms, glutathione, glycine, and other mechanisms also. We have got superoxide dismutase enzyme, we have got catalase enzyme, which act against the free radicals whenever they are produced. But under the influence of cortisol, the production of these antioxidant mechanisms, the production of superoxide dismutase, the production of catalase is decreased. I have told you that cortisol can go inside, can affect the transcription and translation of various proteins. So cortisol does affect the production of superoxide dismutase, does affect the production of catalase. So overall, it decreases the antioxidant profile of the cell. So chronic stress and elevated cortisol can lead to decrease in the activity of antioxidant enzymes such as superoxide dismutase, such as catalase. So these enzymes, normally they help to detoxify the superoxide anion. They help to detoxify the peroxide anion. So reduction in the antioxidant defenses under the influence of cortisol allows reactive oxygen species to accumulate in the body, resulting in inadvertent neurodegeneration. The next and important mechanism is mitochondrial dysfunction. I have told you, mitochondria, they contain highly charged species. So chronic stress can lead to mitochondrial dysfunction, which in turn is associated with neurodegeneration. So mitochondria are the energy producing organelles within this. So they play a crucial role in maintaining the cellular function. Our neuronal cell contain huge amount of mitochondria, active mitochondria. So any disruption of the membrane of the mitochondria will result in all these actively charged species coming out into the neuronal cytoplasm. So chronic stress thus can contribute to the mitochondrial dysfunction. One more very important thing, and it has been recently discovered by Picard in 2015, that chronic stress can lead to changes in the mitochondrial structure also, including elongation of the mitochondrial membrane, fragmentation of the mitochondria at certain points. This can impact mitochondrial function, and it can also affect the energy production capacity of the neuron. See, mitochondria are the organelles which are giving ATP. So if mitochondria are not functioning, reactive oxygen species production will increase, as well as the energy of the cell will decrease. Mitochondrial oxidative stress. So stress can induce overproduction of reactive oxygen species within the mitochondrial membrane. I should tell you, this all happens when in the mitochondrial membrane where electron transport chain is going on. This damages the mitochondrial DNA, proteins and lipids, impairing their function, causing further release of oxidative free radicals into the neuronal cytoplasm. Chronic stress can also result, impair the energy production, biogenesis and dynamics of the mitochondria. It can affect the regulation of mitochondrial biogenesis and dynamics. So dysregulation of biogenesis and dynamics results in depletion of healthy mitochondria. So neurons are there with less energy, impaired ATP levels. So they ultimately go into the pro-apoptotic pathway activation, in which apoptosome is formed, and ultimately the neuron goes into apoptosis. One more phenomenon which has been talked about recently by many researchers is the stress-induced neuroinflammation. Inflammation, we all are acquainted with this term, inflammation. But can stress cause inflammation? See, stress ignites a sustained neuroinflammatory response by activating, I have told you this word, by activating microglial cells and astrocyte cells, which are the housekeeping cells of the brain. Okay, they are also the immune mediator, the immune cells of the central nervous system. When they are mediated under their influence, an immune response is initiated against the neurons. This activation prompts the release of pro-inflammatory cytokines, interleukin 1 beta, interleukin 6, and TNF alpha. I must also tell you, we can include over here a nuclear factor kappa B. This activation prompts the release of these cytokines, and all these TNF alpha, interleukin 1 beta, NF kappa B, they take the neuron from the healthier side into an inadvertent pro-apoptotic path. So slowly but steadily, neuron undergoes inflammation, and inflamed neuron undergoes degeneration. There is energy imbalance, more energy is required, less energy is produced, antioxidant mechanisms go haywire, reactive oxygen species, they pile up. So prolonged stress can result in energy imbalance, and that is why we can see the people who are under stress, they always feel exhausted. They always feel they're having lack of energy. It is not related to their diet. It is not related to their nutrition. It is related to their mental stress level. This phenomenon, excitotoxicity. As the name suggests, toxicity due to overexcitation. It's a well-documented consequence of chronic stress. You are under stress, neurons become excited, neurons become hyperexcited. There is increased depolarization of neurons, increased release of neurotransmitter. And where the neurotransmitter is going to act, it is going to act on the postsynaptic receptors. But there is an equilibrium between excitation and inhibition, equilibrium between excitatory pathway activation and inhibitory pathway activation in the brain. But under the influence of stress, due to overexcitation, this equilibrium is disturbed. This disruption of equilibrium leads to excessive excitotoxic neurotransmitter glutamate being released. Glutamate acts on NMDA receptor and methyl D-aspartate receptors. And overactivation of NMDA receptors results in a phenomenon called excitotoxicity. Excitotoxicity, it has been found out previously also, way back in 1970s and 80s, that it is one of the hallmark features for the development of neurodegeneration. So if we talk about the types of neurodegenerative diseases, I'm just going to talk about three major types. First is the Alzheimer's, which is characterized by accumulation of abnormal protein aggregates within the neurons, such as beta amyloid plaques and tau tangles, tau proteins. These protein accumulations lead to progressive loss of cognitive function and memory. So how this Alzheimer's is linked to stress? Chronic stress has been associated with increased risk of developing Alzheimer's disease and exacerbating its symptoms. Stress-related increase in cortisol, again, cortisol is the culprit, and cortisol-mediated inflammation are believed to contribute to the formation of beta amyloid plaques. Now, this discovery has been made by Yaribeje in 2017. So, being said, okay, stress is involved in Alzheimer's disease. How? Let us see how. What is the mechanism involved? See, number one, chronic stress can lead to overproduction of stress hormones such as cortisol. Elevated cortisol is associated with increased production and release of reduced clearance of beta amyloid protein. Increased production, reduced clearance. This beta amyloid protein forms plaques in the brain of the individuals with Alzheimer's disease. Second, stress can also trigger neuroinflammation, contributing to brain's inflammatory response. So chronic inflammation is believed to play a role in the progression of Alzheimer's disease. Third, oxidative stress. So stress-induced oxidative stress, oxidative free radicals are produced, which cause damage to the cellular components, including the cellular DNA, neuronal proteins, which can exacerbate the production process of Alzheimer's disease. Next, Parkinson's disease. Now, Parkinson's disease is characterized by loss of dopaminergic neurons at specific areas of the brain, nucleostriatal areas of the brain. This results in motor symptoms such as tremor, rigidity, and bradykinesia, slowing down of movements. How it is linked to stress? Chronic stress has been associated with increased risk of developing Parkinson's disease. And sometimes those who have Parkinson's disease, it causes worsening of their symptoms. So stress can really lead to overproduction of reactive oxygen species. We all know that reactive oxygen species, they are the main culprit in Parkinsonism. How? Let us see. Dopaminergic neuron degeneration. Chronic stress can lead to excessive production of ROS, mitochondrial dysfunction. The neurons of dopamine, dopaminergic neurons, they are highly energy producing neurons, highly active neurons. They are very vulnerable to reactive oxygen species. See, the more excited a neuron is, the more susceptible it becomes to oxidative species. Stress-induced neuroinflammation can contribute to inflammatory processes. Again, neuroinflammation, it is much more involved in Parkinson's than in Alzheimer's. Why? Because under the influence of dopamine, there is much more excitation as compared to that of acetylcholine. The third disease I'm going to talk about, which my students have also worked in the lab in 2018-19, is the Huntington's disease. It's a genetic neurodegenerative disorder characterized by degeneration of specific brain regions. Okay, striatum is involved to a major extent. By degeneration of specific brain regions, it results in motor disturbances, cognitive decline, and psychiatric symptoms. So chronic stress can exacerbate Huntington's disease. It can cause oxidative stress and inflammation. Again, both these two are involved. So there might not be a direct link, but it is believed that Huntington's disease can also be non-genetic, environmentally induced. And this environmental induction can be done with by reactive oxygen species mediated and cortisol mediated gene alteration. What are the clinical evidences? See, I have said you have believed this is not the case. So let us see what are the clinical evidences. First finding is that there is impact of chronic stress on Alzheimer's disease. Is there any clinical evidence for that? Chronic stress, particularly when experienced over extended periods, has been associated with the increased risk of developing Alzheimer's disease. Stress-related hormones such as cortisol can lead to accumulation of plaque, beta amyloid plaques, tau tangles, and which are the hallmark of Alzheimer's pathology. What are the studies? See, a study published in Neurology, a famous journal, by Wilson et al. In 2017 found that individuals with a high level of chronic psychological stress had a greater risk of developing Alzheimer's disease. A research published in JAMA, Journal of American Medical Association, JAMA Neurology, in 2013 suggested chronic stress was associated with accelerated cognitive decline in patients already diagnosed with Alzheimer. So you are having the Alzheimer. Again, if people with Alzheimer have chronic stress, there is very fast cognitive decline. Implications. What are the implications of these findings? The findings highlight the potential role of chronic stress in the development of Alzheimer's disease. And they are also, they highlight the importance of stress management in dementia prevention. So if you want to prevent dementia, manage the stress, chronic stress specifically. Then stress and Parkinson's disease. Chronic stress may contribute to the risk of Parkinson's disease by increasing oxidative stress and causing neuroinflammation. So these processes, both these two, oxidative stress, neuroinflammation, they can accelerate the degeneration of dopaminergic neuron. I have told you, dopaminergic neurons are more susceptible than cholinergic neurons. Any proof? A study published in journal Movement Disorders found that exposure to traumatic stress was associated with increased risk in Parkinson's disease. Then a research published in journal Brain, very important, very prominent journal, in 2001 suggested severe life events which often involve significant stress. They were linked to an elevated risk of developing Parkinson's disease. So what are the implications? So these studies, they imply the stress reduction strategy may have a potential role in reducing the risk of Parkinson's disease. Then chronic stress and Huntington's disease. In Huntington's disease, chronic stress can exacerbate symptoms by adding additional burden to the already undergoing neurodegeneration. Stress-mediated oxidative free radicals and neuroinflammation may further damage the neurons which are already affected by the condition. Research published in PLOS One in 2013 suggested chronic stress accelerated the onset and progression of Huntington's disease. A study in Neurobiology of Disease, very famous journal, found that the stress was associated with worsening of clinical outcomes of individuals with Huntington's disease. Again, same as that with Parkinson's. If you are having Huntington disease and you are having stress, the outcomes will be more worsened. Implications. So while these studies, they provide insight into the relationship between stress and Huntington's disease, intervention to manage the stress and improving the quality of life may help with this condition. So what is the impact of stress on the brain structure and brain function? Is it that people who have stress, their brain shrinks? Yes. Let me show you how. The first thing is neuroanatomical changes. So chronic stress has been linked to structural changes in the brain, particularly area like hippocampus, which is critical for memory. Stress can lead to atrophy of dendritic tissue, reduction in new neuron formation, neurogenesis. Okay, so all these processes are associated with brain health. All these processes are hampered by stress. Then neurochemical changes. Chronic stress can lead to alteration in the neurotransmitter levels, dysregulation of HPA axis, and release of stress hormone. They can affect mood, cognition, and overall brain function. Neuroinflammation. Stress can trigger neuroinflammation by activation of microglia and release of pro-inflammatory cytokines. Oxidative stress. Stress can lead to increase in the oxidative free radicals, causing damage to the cellular component, including the DNA of the neuron cells, affecting the protein, affecting the lipids. This damage can contribute to neurodegeneration and cognitive impairment. So we should emphasize on neurodegeneration. That chronic stress, if left as such, not worked upon, not acted upon by any medication or by any other therapy, non-pharmacological therapy, it will inadvertently, slowly but surely, it will result in neurodegeneration. So there is potential importance of stress management and intervention for reducing chronic stress and preventing neurodegeneration. There is neuronal loss. Various brain regions, they lose neurons under stress. But why neuronal loss happens in stress? Chronic stress causes increased reactive oxygen species. The oxidative stress triggers neuronal death through apoptosis, which is a programmed cell death process. There is neuronal atrophy. Neurons, they lose their function, they degenerate. Their structure undergoes degeneration. Okay. But why neuronal atrophy happens? Chronic stress can cause increase in expression of pro-inflammatory cytokines and pro-apoptotic proteins in neurons, resulting in atrophy of dendrites, atrophy of branches of the neurons, degeneration of synaptic connections, and neuronal communications. This in turn affects learning, memory, and other cognitive functions of the body. What is the impact on hippocampus, memory processing area? The hippocampus, or brain's region associated with learning and memory, is particularly vulnerable to effect of chronic stress. So stress-induced neurodegeneration results in structural changes within this region and can result in memory loss. So what happens then? Chronic stress can lead to reduced neurogenesis in the hippocampus, which means fewer new neurons are generated. This in turn impairs memory and cognitive function. It can also cause cortical shrinkage. When I told you, cortical shrinkage, stress-induced neurodegeneration can affect cortical region of the brain, particularly the prefrontal cortex, affecting our mood, affecting our feelings, which is involved in decision making, emotional regulation. All these functions are affected under stress. So it means it shrinks brain. So chronic stress can lead to reduction in the volume of the prefrontal cortex, impacting its cognitive and emotional function. This shrinkage may be associated with alteration in the dendritic branching or alteration in the synaptic connections. Finally, coming on to the point, how can we prevent it? How can we prevent it? Okay, so if stress is a necessary evil, how can we prevent it? Reducing stress and potentially preventing stress-induced neurodegeneration is essential for our mental and physical well-being. Let us see how we can do it. Number one, stress management techniques. Three main important stress management techniques. Mindful meditation. So engaging in mindful practices, meditation, helps us to develop coping mechanisms. Second, yoga. Yoga combines physical posture and breathing exercises, relaxation techniques. This can help to reduce stress and promote overall well-being. Third, this method we can do anywhere, anytime. Deep breathing. Deep breathing exercises can help lower the stress by reducing cortisol level and promoting relaxation. Next, regular physical exercise. Regular physical activity can reduce stress hormones, increase endorphin level, which are natural mood lifters, and improve overall well-being. So aim for at least 150 minutes of moderate intensity per week. Then healthy diet, a balanced diet with adequate nutrients, adequate antioxidants can help reduce the impact of oxidative stress. Adequate sleep, 7 to 9 hours per day. It can help your body regenerate, help your body reduce the antioxidant load, oxidative free radical load in your body. Maintain social connection, not social media, but social connections. Make more friends. Maintain healthy relationship, strong social support network. All these can help to reduce stress. Social stress reduction activities. Engage in those activities which help you to relax, unwind, such as your hobbies, spending time in nature, listening to music. Counseling and therapy. Therapies like cognitive behavioral therapy, CBT, can help you learn effective stress management techniques and develop a healthier mindset. Many teachers would be there in this webinar. They must be talking about this buzzword, work-life balance. Balancing work and personal life, you should give time to both. Don't forget that it is your family who is going to be there with you at the first point when you require. So when you are in the hospital, God forbid if somebody goes into the hospital, the family would be the first, part of the social network that is going to be there with you. All other friends will come late. So we should give time to the families also. Set boundaries. This is my work time. This is my family time. Make time for relaxation, and importantly, generate some time for yourself also. Mindful technology use, less screen time, especially before bed. Less screen time, less scrolling on the internet to improve the sleep quality and reducing the stress. Don't take any thought to the bed. Go stress free. Engage in cognitive training. Talk to more people. Okay. Solve more puzzles. Solve more crosswords. Now, coming on to current research which is going on, research on stress-induced neurodegeneration, which is an evolving field, has very numerous potential breakthroughs. Some of the key research areas are discussed in the next slides. Number one is identifying the molecular mechanisms. How does this happen? Understanding the precise molecular mechanism by which chronic stress contributes to neurodegeneration. So identifying specific molecular pathways. I'm working on them. My students are also working on them. And potential therapeutic targets which can help find a treatment and preventive cure for stress-induced neurodegeneration. Role of epigenetics. Epigenetics means which genetic markers could we identify and target, which can result in our more enhanced stress response, preventive stress response. So research on how stress-induced epigenetic changes can affect gene expression and contribute to neurodegenerative processes, and how we as a pharmacologist can work on these epigenetic changes. Okay, so it can open new avenues to the interventions, to new drugs. Early intervention and prevention. So potential breakthroughs. See, developing strategies for early intervening on the stress-mediated neurodegeneration. The early we work on it, the more successful we will be. So this includes identifying risk factors and creating targeted intervention for each category of stress-mediated neurodegeneration. Then personalized medicine. Tailoring treatments based on individuals' genetic code, individuals' environmental factors. And this can lead to more effective strategy for mitigating stress-induced neurodegeneration. Then gut-brain axis, investigating the relationship between gut microbiota and brain in context of stress-induced neurodegeneration. The gut-brain axis is a very key player in modulating stress response. Next is neuroinflammation and immune responses. Understanding the role of neuroinflammation and neuroimmune response in neurodegeneration is another exciting field in which many scientists from India, specifically from NCCS and NCBS, they are working on. And one of my students in the University of Minnesota is also working on neuroinflammation induced neurodegeneration. Very important lifestyle and behavioral interventions. So how can I change the diet? How can I enhance exercise techniques? How can I manage the stress for preventing neurodegeneration? So we always say prevention is better than cure. So how can stress be managed more effectively with the help of diet, with the help of exercise, with the help of lifestyle changes? so that we may not require any medication for it. So finally, the last topic, implications for healthcare and society. Number one, I kept this on single slides because I want everyone to focus on this. Number one, if there is more stress, more neurodegeneration, it will increase the healthcare cost. So stress-induced neurodegeneration can result in increased prevalence of neurodegenerative disorders, and it can result in long-term care, long-term medication therapy, long-term support services, resulting in higher healthcare cost. Second, caregiver burden. As the number of individuals affected by neurodegenerative diseases increases, the burden on the caregivers, means their wife, their brother, sister, father, mother, anybody, the burden on them, burden on family members also increases. So caregivers, the people who provide care to these type of neurodegenerative disease patients, often experience significant emotional, financial, and physical strain. Workforce productivity. India is a young country. So if young people from our society, they undergo stress-induced neurodegeneration, it will decrease the productivity of the country as such. So chronic stress and its associated impairments can lead to decreased workforce productivity. More individuals experience cognitive decline, there may be less productivity. So it can impact the economy of the country as such. It will have public health impact also, because our public healthcare system is already overburdened. There is one doctor for 3 lakh, more than 3 lakh population in the country. The rise in the stress-induced neurodegeneration, it will put more pressure on the public health system and resources. Finally, the quality of life of the patient. Individuals who are experiencing stress-induced neurodegeneration, they often face a reduced quality of life. Why? Because there is cognitive decline, there is emotional distress, there is diminished ability to engage in daily activities. All these result in reduction in the quality of life. There is a reduction in the psychological well-being, the societal impact. A person who is suffering from a neurodegenerative disorder is considered to be kind of an alien in the society. Only the caregiver comes near to that patient. Other people, they stay away from that person. The stress of caregiving and witnessing the effects of neurodegeneration, it can have huge impact on mental health. And finally, the social support system. We need to develop more social support systems, more social services, more children of school age, college, universities. They should form support groups in order to reduce the stress of their peers. And I'm talking to teachers also over here. Try to reduce the stress of your colleagues. Try to be a counselor for them, so that they do not develop neurodegeneration in later part of their life. So I can conclude that chronic stress can result in neurodegeneration, and stress-induced neurodegeneration has the potential to significantly impact the individual's life. It can impact our country's healthcare system. It can impact our society as such. So we have explored the mechanism through which stress affects the brain, its structure, its function, as well as its potential link to neurodegenerative disease. It's clear now that comprehensive research, comprehensive education, and intervention strategies, they are required. They are the need of the hour. So by understanding the complex biological and psychological and social impact of this phenomenon of stress-induced neurodegeneration, we can open the door to innovative treatments, preventive measures, and a better quality of life to those who are at risk of these disorders. So as I conclude my presentation, I want to say that let's emphasize the importance of stress management. Instead of working on stress-induced neurodegeneration, we should first work on stress management, early intervention, developing support systems to reduce the burden of stress-induced neurodegeneration. So together we can do it. We should work on the well-being of those persons who are under stress. Let us hold each other's hand so as to make a better future. Thank you very much for your patience and listening to me. I'm indebted. Thank you, sir.
Thank you very much, sir. The session is now open for question and answers. So any participants have questions can ask.
Hello, sir.
Yes, sir.
So I have only one question. Actually, at the end, you have explained that there are so many methodology to reduce the stress. So that includes
Yes, sir.
meditation, yoga, deep breathing, regular exercises, and diet, all those different methods. So my question is actually maintaining all these things at once. It is not possible by everyone. So among these
Yes, sir.
different method, any specific method which will give it a better result in reducing stress, can you suggest any one or two
Yes, sir.
which will give the better results?
Sir, first of all, let me tell you that being a pharmacy teacher myself, and you are also a part of that fraternity, it is very right that all the methods we cannot go for at once. But two methods which I recommend, not just in webinar, but also to my colleagues, to my friends, two methods which I refer that everybody can follow is one is deep breathing. This you can do anywhere at any time. Deep breathing gives more oxygen to the brain and helps our brain to combat with the free radicals. Second method, which is again very easy, is 5 minutes of meditation every day. Just 5 minutes. I hope people from all walks of life, all walks, can give 5 minutes to their body. Just 5 minutes of meditation can actually reduce the tension levels to less than 50%. People might be going to their job daily, commuting on various means of communication. We might have experienced that while we are commuting to our job, in between, if it's a half-hour journey, say 15 minutes, 20 minutes, say 1 hour, we sometimes fall asleep. Instead of falling asleep, we can just give 5 minutes to that meditation process. Listening to meditation music. This can be done. Many of the students and faculty members, many of the researchers, they come to their workplace listening to music. This can be replaced by meditation music, just closing your eyes, focusing on the meditation center, the center between the eyes. And most importantly, which I have also talked about, it is to be each other's counselor. When you see somebody in your friend circle, somebody, some colleague of yours is undergoing stress, try to engage that person. Try to engage that person in talk. You can very well make your social circle, but your social circle should be such that they talk to you about their stress. Many a times it has been seen that stress induced pathologies could be prevented only if the person talked his or her heart out to his friends. So make social support group. You can also start in your department. Make social support groups. Try to spend time with them as a faculty member rather than as a principal or HOD. You can start a 10 minute, 15-minute gathering in which social supporting can be done. Believe me, all these small methods, they can prevent neurodegeneration of the future. There is a person who has just raised their hand. I don't know who is. Damodar sir, I want to get that question.
Thank you very much, sir. You gave a very good explanation for this. Uh, I, I requested other participants to raise their questions.
Sir, there is a question in the chat box by our guest, Dr. Sonal Jaiswal. Are there any known mutations in known genes induced by stress that directly correlate to neurodegeneration? She's asking.
Yes, ma'am. Yes. As I've told in the presentation itself, there are a few genes. For example, genes on the BCL-BAX proteins, they are involved. A study by Liu et al. In 2013 and again by his team in 2018 has found out that there are BCL-BAX proteins, and the genes responsible for these BCL-BAX proteins are present on the 7th chromosome of our body, of our genome. So these genes, they become mutated, result in neurodegeneration. Secondarily, in 2019, there was a study by Madrigal et al. Of University of Sao Paulo, in which they have shown that chronic unpredictable stress, I'm working on that, chronic unpredictable stress results in alteration in genes which are responsible for maintaining the striatal health, maintaining the striatal neuronal architecture of the striatum. So they have postulated that these genes which are responsible for striatal health, I'm not getting the name of the particular gene because it was a code actually, they have resulted in, they might result in activation of genes responsible for Huntington's disease. Third is, I'm talking about Alzheimer's disease right now. It has been seen that amyloid beta peptide, amyloid A beta 42, which is majorly responsible for the pathogenesis of Alzheimer's disease, beta 42 production is suppressed by a suppressor protein whose gene is present on chromosome number 7. That gene again can be influenced by environmental toxins, that can be influenced by stress mediated physiological production. And this study was published recently in 2021, I suppose, in Neurological Sciences Journal of Italian Neurological Sciences. Any other question, please, sir?
Good afternoon, Prof. Nitish.
Yes, sir.
Yeah, I am Dr. Shabi.
Hello,
I'm an
sir.
academician of this institution. I just wanted to express my sincere gratitude for your presentation. Your contribution was valuable in the topic of stress-induced neurodegeneration. That insights provoking the discuss among us. I feel that I travel from K to C, that is kindergarten to college program. That much clarity was there in your PowerPoint. It's really captivate information. I just have two questions. You have covered very clearly introduction, understanding the stress, neurodegenerate basics, stress and brain related things, biological mechanism, biochemical mechanism, type of neurodegeneration, and clinical evidence. Of course, this morning I just read after seeing your presentation. Just read the recent publication is related to it. It has been published 19th October 23. Is a genetic architecture of cerebral ventricles and their overlap with the neuropsychotic traits. Since a lot of some students are there, we are midst of this project. I'm wondering if you don't mind, can you share some few research which will be useful for our PG students? Many students are here. And can you please share what are the project is going on under you related this?
Okay, no problem, sir. As far as students are concerned, I would like to tell the students that they should target, because it is not the era or the time in which the supervisors, they tell the students to work on a particular molecule or a particular disease, because now the time has changed, and you will agree with me that students are much more sharper, having more sharper brains than us. Yeah, where I can guide the students is that there are various downstream pathway proteins of neuroinflammatory cascade. There are right now those scientists who are working on neuroinflammation. There are around 15, if not more, around 15 neuroinflammatory neuroinflammatory processes which are being explored all over the world. These cascades, each cascade has around 10 to 15 proteins, and in those cascades there are certain common proteins. Common proteins means where different branches of the cascade, they coalesce together. For example, in NF-kappa B mediated cascade, there is a protein P65. Now this protein is the differentiating factor between the physiological and pathological cascade of NF-kappa. How does a physiological protein becomes a pathological protein? How a conformatory change takes place inside the P65 in order to become NF-kappa B from take it from physiology to pathology. If I can do docking studies, molecular modeling studies of my proposed drug with this protein, and if I want to inhibit the protein, and if the docking score is a negative, so prior to initiating the pharmacological studies, prior to initiating the in vivo studies, I should go for docking my molecule with one of the downstream proteins of a neuroinflammatory cascade. Then, if the results are fruitful, you will find physiological action. You will find the pharmacological action. I have worked on recently on one of the molecules, that is androgaffolide, and we have found wonderful results. And your second question, which you have told me that how we should go ahead in this field of stress-induced neurodegeneration. So first thing is we should work on developing new models of stress. The existing models of stress, they are inadequate. We should work on developing new models of stress. This could also be one of a good project for your research students. And we should also go for the molecular cloning of the genes responsible for stress, the genes which I have already pointed out in answer to one of the questions. If you have the facility, or if you are willing to collaborate with some of the institutes of eminence, you can go for PCR cloning of the genes responsible for stress. These are the two methods which right now I can explain.
Prof. Nitish, I have one more question that it may be related to Dr. Anbu's question. Dr. Anbu asked specific, non-pharmacological therapy, something like that. Actually, these are the in your PowerPoint says it's very clear, especially for the practice. So since it's old age people, of course, the abroad or the developed country, there is a different session for this type of practice. Is there any option for providing the training program for the old age people who are stress-induced neurodegeneration?
Yes, sir. I must correct you right now over here that this stress-induced neurodegeneration is not a concern for the elderly. Even those who are undergoing neurodegenerative disorders, it might be that their neurodegenerative disorder might be age-induced or any other. Stress-induced neurodegeneration is a problem or concern, I should say, for the young population. Why? Because stress-induced neurodegeneration is accelerated form of neurodegeneration, which occurs in the young age. So, as you have said, we should have training programs, definitely. But those training programs should be targeted at all of the population, starting from 15-year onwards. Even students who are 15-year-old, 14-year-old, 13-year-old, they are under tremendous stress because of the competitive nature of the work environment that we are facing here. Education environment and as well as the work environment, it has become very competitive. Because of this competition, we are facing stress. So these training programs should be there, definitely should be there, but they should include all of the population. Anybody could be stressed, and anybody who is stressed for a longer period of time and is not able to cope with that stress is, we can say, is amenable to stress-induced neurodegeneration.
Thank you, Prof. Nitish. Actually, I like the thing related work-life balance.
Yes.
We need it actually.
Definitely.
Okay. Again, we are deeply grateful for your participation. We hope to have opportunity to work with you again in future. Thank you, Prof.
Thank you. Thank you, sir.
Good afternoon. Ms. Nitish, can you hear me?
Yes, ma'am.
Yeah, thank you very much. It's a wonderful talk. Really, I enjoyed it. Yeah, I totally agree with you that this particular stress degenerative is for all the age groups. That's my part of telling you. And I just have one question for you.
Just a second, ma'am. You are not audible clearly right now. Dr. Sunita has raised her hand. She's not audible in the group.
Can you hear me, doctor?
Not in the group.
No, it's actually her voice is a little feeble and there is some echo. Madam, can you please raise your voice a little bit high?
Yeah. Can you hear me, sir?
Little loudly.
Now is it
Yes.
Yeah.
Yes, ma'am.
Yeah. So my question is, can you suggest any in vitro cell line models for neurodegenerative disease?
Yes, ma'am. There are a few in vitro cell line models for neurodegenerative diseases. If I'm talking about neurodegeneration as such, I'm not talking about stress-induced neurodegeneration. Then there are a few models of for neurodegenerative disorders. But if I'm talking about stress-induced neurodegeneration, because stress is a subjective phenomenon, we need to put the animals under stress, then neurodegeneration will start. Secondarily, there are a few models that were developed by one of my friends in Massachusetts University, MIT, Massachusetts Institute of Technology, in which they took out this developing dopaminergic neurons from the fetal cell line, and they cultured those fetal cell line neurons. And while they are being cultured, because they are fetal cell lines, so they will develop over a period of time. So those fetal cell lines, while they are being developed in vitro, they were then put under an external stressor by giving them a modified cortisol into the cell culture. So this modified cortisol, although given in very minute quantity, picomolar quantity, it produced uh, the stress response in the developing neuronal culture in the same way as that would have happened if that would have taken place in vivo in the animal body. Then in the drug treatment groups, they pre-treated the cell culture with one of the prominent antioxidants, that is vitamin C, in the cell culture, vitamin C again in the very low quantities. And then under the influence of vitamin C, the effect of modified cortisol, it was a semi-synthetic derivative of cortisol. The effect of that semi-synthetic derivative of cortisol was reduced to nearly 30% of what it was in the control group. But this stress-induced neurodegeneration in vitro model is being developed in the MIT, and this experimentation took place in 2020-21. So we might see stress-induced neurodegeneration in vitro model in the near future in the textbooks. And if we talk about neurodegeneration models as such, I'm not talking about stress-induced neurodegeneration model. If we're talking about neurodegeneration models as such, yes, we have got neurodegeneration models right from 2001. We have got neurodegeneration models. First model is the dopaminergic cell line model, in which dopaminergic cell lines are used, and this model was developed by Biocen company, and it is available as a kit. Second model, which is most commonly used by researchers worldwide, is the in which neuroglial cells are used. Neuroglial cells are used, and this in this model, neuroglial cells are then they are made to interact with cortisol derivative. This is again given as a standard in the kit, and the progression of neuroglial cells, the extent to which neuroglial cells they develop on the culture plate, the area and the surface density, the density of the glial cells over a period of 48 hours is measured. If there is a reduction in the density, if there is a reduction in the redox potential, because a redox sensor is also there, then that is considered to be stress response. So there are a few models for neurodegeneration, but for stress-induced neurodegeneration, one of the models is being developed by MIT.
Thank you, Dr. Nitish.
Thank you, ma'am. Any other question, please?
Participants can unmute your mic and you can raise the question directly.
And if any other questions are there, I request the participants to put their questions in the chat box, and it will be answered by the speaker. Now I request Ms. Tanuja, Assistant Professor, Faculty of Pharmacy, Ramaya University of Applied Sciences, to deliver the vote of thanks.
Distinguished colleagues, participants, now we are streaming towards the end of today's webinar. It's my honor and privilege to conclude this session by extending a vote of thanks. First of all, we are very much thankful to the management and inspirational source, Honorable Vice Chancellor Dr. Raina and Pro Vice Chancellor, for their continuous encouragement and support in conducting webinars. On behalf of all the faculty members of Department of Pharmacology, I thank our Dean Dr. S. Bharat for his all-round contribution to the Faculty of Pharmacy with his proactiveness and valuable guidance. And we are really fortunate to have an eminent speaker of today's webinar, Professor Nitish Bhatia sir. In spite of his busy schedule, he agreed to deliver this talk, and we are very much thankful to you, sir. In this discussion, he emphasized about various body responses involved in acute and chronic stress, physiological responses to stress, and how stress affects neurons and results in neurodegeneration, mechanism of cortisol in production of reactive oxygen species, and mitochondrial dysfunction, and various other mechanisms. And his enlightening session is definitely useful for the young researchers in our field of pharmacy. On behalf of Department of Pharmacology, Faculty of Pharmacy, and all other participants, I profusely thank Dr. Nitish Bhatia sir for his thought-provoking session. I would also like to convey my sincere appreciation to Mr. Damodar Naik, Assistant Professor, and our department faculty members for their support and cooperation throughout the process and arrangement to successfully conduct this webinar. On my personal behalf and behalf of Department of Pharmacology, Faculty of Pharmacy, I profusely thank all the participants for their active participation, support, and presence in making this program a noteworthy success. Thank you, one and all.
Thank you,
sir.
Nitishar. We'll meet you again for some other session. So very soon we will have one more session in detail about a similar kind of topics.
Thank you, sir. Thank you.
Thank you, one and all. You can leave the meeting now.